Thank you. So yeah, I'll be presenting my work on SYNGAP1 in endothelial cells. So first I'll give you guys all a background on the vasculature. So that's a main focus in our lab. Okay, here we go. So your brain consists of 2% of your total body mass, but it takes up about 20% of the energy that's produced by your body. So it's a highly demanding organ, and it has very limited energy supply. So we need this constant supply of nutrients and oxygens to the brain, so your neurons, for proper functioning. So if you lose vasculature function, you also get a dysregulation in neuronal function. And so your vasculature is made up of multiple cell types. So there's endothelial cells, which are in pink, and they line the blood vessel wall, so they're the building blocks of your vasculature. They're surrounded by pericytes as well as astrocytes in purple, which connect to the neurons in blue. So we know a lot about, I'll briefly go over some of the symptoms in ASD, but it results in intellectual disability, hyperactivity, repetitive behavior, and deficits in social interaction and communication. And this can be caused by environmental factors as well as genetic factors, as we know. And what we've previously been looking at in the lab is copy number variation, so 16p122 deletion, 11.2 deletion. And so I'll talk a little bit about that and how that's implicated in the vasculature, and as well as my project on SYNGAP1. So a lot of the focus in ASD as well as other neurodevelopmental disorders has been on neuronal function. But there is increasing, there we go, like that. There is increasing evidence of the vascular contribution to ASD. So studies in humans have shown that there is vasculature impairment in neurodevelopmental disorders. So a study showed that in patients with ASD, there is altered cerebral blood flow, as well as dysregulation in cerebrovascular structure in a postmortem study. And we're able to model this as well as in mice. So when you delete a ASD-linked gene specifically in endothelial cells, it can result in motor impairment, as well as the deletion of endothelial SHANK3 led to BBB permeability, so increased BBB leakiness, and also neuronal dysfunction. And this was in neonatal mice. So our lab has also looked at the 16p11.2 deletion, and we see that there's early endothelial dysfunction that led to behavioral deficits later on. And this was shown by a delay in vascular development. So as you can see here, at postnatal day 14, which is a critical time for vascular development, there is a lack of vascular density as well as branching. And by postnatal day 50, which is about adulthood in mice, they catch up. However, there is impaired neurovascular coupling, so the response of your blood vessels to neuronal activity. So here we see a delay in that response, shown here by a reduction in cerebral blood flow once the neurons are stimulated. And then when you isolate endothelial cells from these mice, you see a reduced vascular growth in vitro. And so this is shown by a lack of ability to form these networks, as you can see in the wild type. And then interestingly, this was actually conserved to human samples. So from iPSCs derived from patients with the 16p11.2 deletion, they also aren't able to form these in vitro vascular networks. And so what we would like to know is if this can be generalized to other neurodevelopmental disorders, and that brings me to SYNGAP1. So as we know, it's a rare genetic de novo mutation and accounts for about 1% of ID cases, and it has multiple symptoms: intellectual disability, seizures, speech and language problems, as well as motor problems. And at a cellular level, we know that it increases synaptic activity, results in cytoskeleton dysfunction, as well as neuronal dysfunction. However, it is present we know a lot about the function of SYNGAP1 in neurons, but we don't know its role in other cell types. So SYNGAP1 is expressed in other cell types, so in astrocytes, in oligodendrocytes, as well as in endothelial cells. And this is from single cell data in mice. And then from my own data set looking at RNA, in human-induced endothelial cells, we do see an expression of SYNGAP1. And so we know that SYNGAP1 regulates the RAS-ERK pathway, and ERK is a key effector of VEGF, which is important for endothelial proliferation, as well as vascular growth. So what we would like to know is how is SYNGAP1 contributing to the structure, function, and maturation of endothelial cells? So my hypothesis is that the haploinsufficiency of SYNGAP1 leads to cerebrovascular changes and function. And this will be looking at in vitro and in vivo models to determine this. So my first aim is to assess the impact of SYNGAP1 haploinsufficiency on endothelial cell molecular signatures, structure, as well as function in vitro. So through Cure SYNGAP1, we obtained SYNGAP1 patient-derived induced pluripotent stem cells, and here we have 8 different mutation cell lines, each at different locations on the gene, as well as different types of mutations, and they also have sex-matched controls. And so with these, I'm able to generate human-induced endothelial cells. So the cells are cultured in an iPSC media, and then they are changed to a media that will promote proliferation, prime them for differentiation, and then finally they're supplemented with VEGF, which will induce them to turn into endothelial cells, and then I can select these cells and have a pure culture of endothelial cells. And then just to confirm that we do indeed have endothelial cells and other cell types in the culture, we looked at the RNA expression and we see an enrichment in endothelial cell-specific genes. So with this, our first step was to characterize these cells, so by staining them with V-cadherin, which is an endothelial adherence marker and is very important in blood-brain barrier function. We see when we look closer, actually, there is a drastic difference between the proband and the control line, where in the proband, you see an increase in number of gaps along the membrane. So here this graph is showing 50 cells per cell line. And we see also a decrease in the thickness of the V-cadherin expression along the membrane. So here we're showing that there is altered cell-to-cell junction in SYNGAP1 patient-derived human-induced endothelial cells. And then when we want to assess vascular function, we can seed these cells into a artificial matrix that will promote them to form capillary-like networks. And then we can image this at 2 hours, 4 hours, 6 hours, 8 hours, and 24 hours, and assess how they're able to form these networks over this time course. So in our first cell line here, we see that actually the proband has an increase in this capillary-like formation at 2 hours. However, by 4 hours, it collapses compared to the control. And as you can see in this picture, it's a representative image of the 4-hour time point. And then in the next cell line, we actually see an overall overgrowth of vascular formation in these cells. And then in our last cell line here, it's completely different. We see a lack of formation. So these SYNGAP1 patient-derived human-induced endothelial cells do display abnormalities in their ability to form networks, and it differs from mutation to mutation. And then what we did also was to isolate RNA and assess their differentially expressed genes. So in this PCA plot, what you're seeing is how the cell lines will naturally separate. So in purple, you have the controls, and in red you have the probands. And you can see that they separate, but also at the top you have a few cell lines that are separate from the others, and those are actually females. So there might be some sex-specific differences here. More analysis remains to be done. But overall, when you look at all the controls and all the mutations together, we see that there's 325 upregulated genes and 118 downregulated DEGs. And then when we look at controls versus probands, we can see that there's 2 distinct gene clusters that are driving these differences. And then also looking at a volcano plot, we can see the top differentially expressed genes, and a lot of them are related to vascular regulation. So the ones that are in red here that are upregulated are involved in promoting vascular maturation as well as in proliferation. And some of the downregulated genes, like MAOX2, is actually involved in endothelial quiescence. So what we're seeing here is that SYNGAP1 does impact endothelial gene expression quite significantly, and we'll be doing more analysis on each of these genes as well. So my second aim was to determine how SYNGAP1 shapes neurovascular development and function in vivo. So I'll be assessing cerebrovascular structure, function, and behavior. But just for today, I'll give some very preliminary results we have. So we're using an endothelial-specific mouse model. So we delete SYNGAP1 specifically from endothelial cells in mice, and we're able to have a homozygous as well as a heterozygous deletion of SYNGAP1. And so with these mice, I stained them for CD31, which is another adherence marker in endothelial cells. And this was done at postnatal day 50, so young adulthood. And we see a decrease in the average vessel area coverage in adulthood for mice. And then also what we did, we're also assessing cerebral blood flow. And so far there's no differences, but we do see a trend towards an increase in cerebral blood flow at postnatal day 50 as well. So the take-home message is that SYNGAP1 doesn't only affect neurons, and that SYNGAP1 alters endothelial expression, junctional structure, and angiogenic behavior. And this is highlighting a new perspective for SYNGAP1 and looking at the vasculature. So I'd like to thank Cure Syngap, as well as the families that support this research, and my lab members. And we two that have helped me along the way are Dr. Mo and also Amber Morton, who's an undergrad doing the behavior study for me. So thank you very much.